Hi, good evening, guys. Just let me know if you can see me live, if you can see me, my live feed. Okay, so I'm live. I can see myself uh, you- live on YouTube. So let's start today's session. So today's session again is a very important topic on carbohydrate metabolism, that is glycogen metabolism. Now, I will be talking only about the glycogen metabolism in this session and not about glycogen storage disorders because that will be a separate, session altogether. So for anyone who is attending this session for the first time, I'm Dr. Abhishek, your mentor at Medicoapps, and I try to take a lot of live sessions at 11 p.m. On high topics, especially from biochemistry and clinical topics from internal medicine. So welcome to this session, guys, and these are the sessions which I have taken in past 3 days. I've taken a session on glycolysis on the 19th, gluconeogenesis, then electron transport chain. All these are very high yield topics, and today I'm taking this session on glycogen metabolism. Again, next week's sessions I will announce as they are, planned. If you are new to this session, if you are new to Medicoapps, make sure you subscribe. If you like this session, make sure you like it so that it gets saved in your YouTube history. If you have any question to this session, or if you have any suggestion as to what kind of topics can be taken on live sessions, you can just put in the comment. If you have any question, if you put it, I'll reply to it. And if you really like this session, I would request you to share it with your friends. Getting started with glycogen metabolism. So what is glycogen essentially? Glycogen is a polymer of glucose. Okay, so glucose, multiple molecules of glucose joined together. So it's a polymer of glucose, and it is primarily used as a storage form in our body. So glycogen is a polymer form of glucose, and it is used as a storage form of glucose inside the body. Why do we need to store glucose? Now we know that whenever we are into a fasting state, that is in between meals, the glucose level will start dropping down, and there are certain organs which exclusively depend upon glucose. There are certain organs, especially 5 organs you have to remember. Remember, you have to remember brain exclusively depends on glucose. Your RBCs exclusively depends upon glucose. Your lens and cornea again exclusively depend on glucose. Your renal medulla, and lastly, if you're a male, your testis. Okay, so these organs exclusively depend upon glucose. So if in between meals, if the glucose level starts fall down, this is the first defense mechanism of the body to not allow the glucose levels to fall down. Now typically, a question is asked is how long will glycogen stores last in our body. So there are two organs where this glycogen is stored, and we talk about liver and muscle are the two most important organ for storage of glycogen, and it is the liver glycogen. We are going to see the difference between liver and muscle glycogen in a while, and it is this liver glycogen which is the major storage form, and typically 16 to 18 hours, okay, is what will be a typical glycogen store that will last. So after overnight fasting, your glycogen store will almost be depleted, and this has a lot of consequences when we talk about fed and fasting state. A session on that, we'll talk about it. But remember, your liver glycogen stores can last only up to 16 to 18 hours, okay, which means after overnight fasting your liver glycogen is almost nil. Now, having said that, a lot of questions in examination will be asked between the difference of liver glycogen and muscle glycogen. Okay, so what is the essential difference between the glycogen which is stored in the liver and the glycogen which is stored in the muscle? Now, remember, liver is like mother. Okay, liver is like mother, which means whatever glycogen is stored in liver, whenever the blood glucose level falls down, it will convert it into glucose and you know give it to the blood so that it can be used throughout the body. So very selfless, whatever it is storing, it is purely storing for the entire body. Whenever the blood glucose level falls down, this glycogen will be broken down, and all that glucose will be pumped into the into the blood so that the entire body can use it. Whereas muscle is very selfish. Okay, muscle is selfish, which means whatever glycogen is stored in the muscle, whatever glycogen is stored in the muscle, the muscle will use only by itself. It will not give it to the entire body, because muscle will convert glycogen into glucose 6 phosphate, and we know that glucose 6 phosphate cannot exit the cell, and that is why whatever only by itself. Okay, so whatever glucose is being stored by the muscle, it will only convert it to glucose 6 phosphate and use it by itself. So this is a very, very important difference that you have to remember. So first difference, whatever glycogen is stored in the liver, it is available for the entire body. Whatever liver is stored, glycogen is stored in the muscle, it is only available for muscle. The second big difference is, if you see the glycogen level in liver, it is fluctuating like this. It is fluctuating like this. Okay, which means the glycogen will increase, decrease, increase, decrease. And what is this increase, decrease dependent on? Remember, it increases when you are in fed state. It increases when you are in fed state. So whenever you eat, your liver glycogen will increase, and it decreases when you are in fasting state. It decreases when you are in fasting state. Okay, so which means liver glycogen is dependent on the fed and the fasting state. Any doubt in this? Liver glycogen is dependent on the fed and fasting state. Let's talk about what about our muscle glycogen. Again, when we look at muscle glycogen, okay, so this is liver glycogen. Again, look at muscle glycogen. Again, muscle glycogen will increase, decrease, increase, decrease with time. But here, if you see, the muscle glycogen will increase when you are in rest state. When the muscle is in rest state, okay, the muscle will accumulate more glucose inside it and convert it into glycogen, and it will decrease in your exercising state. Okay, so again, the level of glycogen in muscle keeps on fluctuating, but that fluctuation, as I have told you, is dependent upon the rest and exercising state. Okay, so this is the second major difference between the glycogen which is stored in the liver and the glycogen which is stored in the muscle. And the last difference which you have to remember, the third difference which you have to remember, is liver has higher density of glycogen storage. Okay, we see typically liver has a weight of around around 100 grams of glycogen is stored, which is almost 10% of the weight of liver. Liver is around 900 to 1000 grams. So almost 100 gram of glycogen is stored in the liver. Okay. Whereas muscle has lower density. Okay. If you see muscle, around 400 grams are stored, which is 1 to 2% of the muscle. But interesting point here is the total content of glycogen in muscle is much more than that in liver. And this is the point they will trick you in the examination. So they will ask you, where is more glycogen stored? Is it in liver or is it in muscle? The total amount of glycogen which is stored is more in muscle, because muscle has around 400 grams of glycogen stored, whereas liver has only 100 grams. But the density of glycogen storage is higher in your liver, because almost 10% of the liver is glycogen. Okay, whereas 1 to 2% of muscle is glycogen. So these are very, very important factors. So let's look at one by one. As I have told you, in liver, it is stored at higher density, almost 10% of the weight of the liver, but in muscle, it is stored in larger quantity, but the density is less. Second, I have told you, glycogen is changed to glucose in liver and enter into the circulation, but in muscle only it is converted to glucose 6 phosphate. And the last is, in glycogen in liver is dependent on fed and fasting state, whereas glycogen is in muscle is dependent on rest and exercising state. The last important difference is why is this difference between liver and muscle? It is because of the glucose receptor. We know that liver has GLUT2 receptors. What is GLUT2 receptors? It will allow bidirectional flow of glucose. So suppose I'll draw an example so that you are clear. Suppose this is a liver cell, okay, and this is your GLUT2 receptors. Okay, this is your GLUT2 receptors. So whenever the density of blood glucose is high, so it will allow the extra glucose to get into the liver, and whenever the blood glucose falls down, it will allow the glucose to exit. So this is the meaning of bidirectional. So liver has GLUT2 receptors, okay, and it is always present on the ear. Whereas suppose this is a muscle cell. Let's draw a muscle cell. So let's say this is the muscle cell, and it has, now the muscle cell GLUT4 receptors, they will never be on the surface. They will always sit inside the cell. Whenever the blood glucose level is high, then it will migrate to the surface, and it will suck in all the extra glucose. The moment blood glucose level falls down, it will again go and enter into the into the cell. So essentially, these GLUT4 receptors, which are present in the muscle, they allow only entry of glucose, not exit of glucose, and that is the reason why it is. Hi Bio Ninja, I'm Go Trio. Okay, so this is about the difference between liver glycogen and muscle glycogen. I hope everyone is clear. Just a small reminder, I send daily updates like this session or any live session. We send daily high yield topic updates in terms of video lectures, quizzes, concept cards, a very interesting way to revise. And all that you can get it by joining our free WhatsApp group. They are only limited groups we have formed only for serious students. And the best part about that group is that nobody else can post except me. So it's not no good morning, good evening messages, only high quality update. And to join these groups, all you have to do is go to www.medicoapps/groups. Okay, medicoapps/groups. So if you search for Medicoapps group, and you can join whatever group you like. So having understood that, now let's look at what is the structure of glycogen and how glycogen is formed and broken down. So this is the structure of glucose. We know this is the first carbon, this is the second carbon, third carbon, this is the fourth carbon, this is the fifth carbon, this is the sixth carbon. Everyone is clear about the structure of glucose. Now let's look how glycogen. We know that it's multiple glucose joined together. So there are two different kinds of bond. So one is 1,4 bond, we call it, which is the first carbon here and the fourth carbon here. This will form a bond. Okay, so this is called as an alpha 1,4 glycosidic bond. Another bond is first carbon here and sixth carbon here. So this is called as alpha 1,6 glycosidic bond. So there are two kinds of bond. Let me give you a diagrammatic explanation so that you are more clear. So this is your glucose. Let's convert it into a man. So 4 is head, 1 is leg, and 6 is your arm. So when I talk about, so if we rotate it, so this is how it will be. When I talk about 1,4 bond, so it is like one glucose standing on the top of other, one glucose standing on the top of other. And when I talk about 1,6 bond, you can see, so it is like 1,6 bond is there. So it is like it's a branching point. You can see very easily it's a branching point. I hope all of you are clear about this. See, it's a branching point. So 1,4 is one glucose on the top of other, so it's a linear arrangement, and 1,6 is like a branching point in a tree. Okay, so if I have to make a diagram, it will look something like this. It will look so. This is your 1,4 bond, one glucose sitting on the top of other, and this is your 1,6 bond. And again, on the top of other, it is 1,4 bond. Again, if there is a branching point, this is called as 1,6. Okay, this is called as 1,6. So this is all 1,4, 1,4, 1,4. This branching point is 1,6. This branching point is 1,6. And it is very important for you to understand the structure of glucose, right? So all of you are clear about the 1,4 bond and the 1,6 bond. Now let's look at quickly the glycogen synthesis, how glycogen synthesis happens in our body. So it all starts with glucose. Now glucose has to enter into glycogen family. Glycogen family is a rich family. It's a sophisticated family. So glucose has to, so that yes, it also has some power within it. So it has to get activated. And how does it get activated? The first is basic glucose getting converted into glucose 6 phosphate. So I told you 6 is the R position, so it is keeping a phosphate in its R position. There are two enzymes, glucokinase working in the liver and hexokinase working in the muscle. Now, as I told you, this glucose 6 phosphate, when I discussed about glycolysis, this can go into variety of pathways. It can go into your glycolysis. Okay, it can go into your HMP pathway. Yes, it is already a part of glycogen synthesis. Okay, and then again it can go into uronic acid pathway. There's multiple pathways in which this glucose 6 phosphate. So here we are going to study about how it will get converted into glycogen. So then what will happen? Now this, anyone can put a power bomb in hand, but this glucose has to demonstrate that it is very powerful. So how it will do? It will move this phosphate from leg position to from arm position to leg position, which means from glucose 6 phosphate it will get converted into glucose 1 phosphate. The enzyme here is phosphoglucomutase, and magnesium is the catalyst here. Again, this is a very important exam. I'll tell you what kind of question is asked on this enzyme, but just be with me. So this glucose 6 phosphate is converted into glucose 1 phosphate, but still it has to show more power. I told you this glucose has to enter into the rich family of glycogen, so it has to demonstrate power, right? So this glucose 1 phosphate will now combine with another rich molecule, that is UTP. We know UTP, GTP, ATP, all these are energy rich molecule. And what it will do is it will remove 2-phosphate and take and get converted into UDP glucose, which means there are 2 phosphates and glucose. Okay, UDP plus glucose. Now this UDP glucose is an activated glucose. Again, a very favorite question. What is activated glucose? So remember, activated glucose, a very important MCQ. What is activated glucose? They will ask you. Remember, it is UDP glucose. And as I have told you how it is formed, it is formed when glucose 1-phosphate reacts with UTP. Okay, we know all UTP, ATP, GTP, all these are high range. Now this UTP glucose is now ready to go into the glycogen family. So how does it happen? So basically this is glycogen primer. Now remember, liver at the time of birth only has some glycogen structure that is called as glycogen primer. So no enzyme is involved in formation of primer, it is already pre-existing. But let's say this is the new glucose which comes. The first enzyme which will be helping is glycogen synthase, and what it will do is it will simply add glucose residues on the top. So it is creating 1,4 bond. So glycogen synthase, the first enzyme, is creating 1,4 glycosidic bond. One glucose on the top of other, one glucose on the top of other. This is the function of your glycogen synthase. Obviously, when 10 to 12 residues are added, glycogen has to have a branch structure. There was a question why glycogen is having branch structure, because two things. First of all, if we see branch structure, more amount of glucose we can store it inside a single cell. And second is, if you have a single, let's say glucose is having only a single linear structure, at one time of breaking only one glucose can be removed. But if it has multiple reducing ends, okay, at one time 3 glucose, or the more number of reducing ends will be there, more glucose can be released at the same time. So these are the two reasons, but the more important reason is that a branch structure allows for a more compact arrangement of glucose. So this glycogen synthase is only doing a linear arrangement, it is just adding here. Imagine now the next enzyme comes, that is called as the branching enzyme. Now branching enzyme does two things. First of all, it will cut the, once the 10-12 residues are added, it will cut it, and then it will create a branching point by 1,6 bond. So it has 1,4 glucosidase activity. Remember, here it is glycosidic, which means it is creating a 1,4 bond. This branching enzyme is cutting the 1,4 bond and creating a 1,6 bond. So now you are having a structured glycogen. Anyone? Okay. So multiple, so is everybody clear about this? BioNinja asked me a question about multiple fates of glucose 6-phosphate. So let me talk about the multiple fates of glucose 6-phosphate. Remember that the conversion of glucose to glucose 6-phosphate, it's a mechanism to trap glucose. Let me explain this why. Suppose this is the cell, okay, and this is the cell, and let's say a glucose enters into the cell. The point is that this glucose can easily exit also, because this is a non-polar molecule. It can easily enter the cell and it can easily exit the cell. What if the cell, to keep the glucose inside, converts it into glucose 6-phosphate? Now this becomes a polar molecule and your glucose channels cannot let it out. And that is how the glucose is trapped inside the air. In fact, all the energy rich molecules, if you see, fructose is converted into fructose 1-phosphate, galactose is converted into galactose 6-phosphate. So any energy rich molecule will be, phosphate will be added so that it gets trapped. And that is why this step is called as trapping of glucose. Okay, trapping of glucose. Now what can happen to this glucose 6-phosphate? It can go into multiple pathways. The most important pathway being glycolysis. Okay, then the next important pathway is HMP shunt. Okay, almost 5%, 10% glucose goes into HMP shunt. Then uronic acid pathway. Okay, next is your glycogen synthesis. So these are the important pathways in which your glucose 6-phosphate can enter into it. Yes, you are very correct that number of branching point will decrease as we are moving. Okay, so obviously as we are moving from the center, the number of branching points will decrease, because obviously the length also has to increase. So this is glycogen synthesis. Let's quickly look at glycogen breakdown. So here is your the first enzyme to act on glycogen breakdown is your glycogen phosphorylase. Okay, the first enzyme is glycogen phosphorylase, and it simply cuts the 1,6 bond, 1,4 bond. Okay, so it is reducing the length. The limitation of glycogen phosphorylase is it cannot cut the last 4 bond. So let's say this this is the first, this is the second, this is the third. So last 4 residues, this first, second, third, fourth, 4 residues or last 3 bond it cannot cut. So it will be able to cut this, it will be able to cut this, but it will not be able to cut this bond. Similarly, if you see 1, 2, 3, 4 residues. So it can cut this bond, it can cut this bond, it can cut this bond, it will not be able to cut this bond. Okay. So the glycogen phosphorylase can cut only up till the last 4 residues. The last 4 residues it cannot cut. Then what happens? We'll see how the last 4 residues are cut. But when glycogen phosphorylase cuts, it does not produce glucose. It produces glucose 1-phosphate. And this glucose, because if it produces glucose, it will quickly move out of the cell. So it produces glucose 1-phosphate, and with the help of phosphoglucomutase, this glucose 1-phosphate is converted into glucose 6-phosphate. And then what happens? There is an enzyme called as glucose translocase, and it transports this glucose 6-phosphate inside the endoplasmic reticulum, and where this glucose 6-phosphatase enzyme is there, which will remove the phosphate and convert it into free glucose, and this free glucose can exit the cell. So this is the mechanism. I told you that in muscle only glucose 6-phosphate is formed, not glucose, pure glucose, because muscle cell lacks this glucose 6-phosphate. Is it clear? Muscle lacks this, so in muscle only glucose 6-phosphate can be formed, and because I told you that glucose 6-phosphate cannot escape the cell, so that is why whatever glycogen is stored in the muscle, it will only be converted into glucose 6-phosphate, and it will only be available for the muscle. Is it clear? Now, as I have told you, let's look at what is the debranching enzyme function. Okay, now debranching enzyme has 2 functions. One is glucose transferase. So what it will do is, so last 3, so 1, 2, 3. Okay, so what it will do is, except for the last residue, the last 3 residues it will transfer to the nearest branch. For example, let's say this is the case. Let's see this case clearly. These 4 residues now can no longer be acted upon by glycogen phosphorylase. I told you that 1, 2, 3, 4. Last 4 residues cannot be acted upon by glycogen synthase. Now what happens here is, when the glucose transferase or the debranching enzyme will come, what it will do is take the 3 residues from here and transfer it here. So what is going to happen? So once 3 residues are transferred here, now again your glycogen phosphorylase can cut these 3. And what about this? The same debranching enzyme also has 1,6-glucosidase activity, so it can directly cut. And one question they will ask you is, again, if this bond is cut, which means 1,4 bond is cut, only glucose 1-phosphate. But whenever 1,6 bond is cut, directly free glucose is formed. Is it clear to everybody? Yes, Bioninja, it is very clear. Good question. Muscle cannot regulate the blood glucose. Okay, muscle cannot regulate the blood glucose. It is only your muscle can regulate actually the blood glucose, not through glycogen, but what happens whenever you eat. Okay, that's a very important question. Let me answer that question about role of muscle. Role of muscle in glucose metabolism. Okay, now we told that obviously glycogen will not contribute to anything in the blood glucose. But muscle has a very important role, and let me tell you what. The moment you eat food, okay, there is huge amount of there is increase in blood glucose. What will happen? This blood glucose will increase insulin, and what will this insulin do? It will, in a muscle cell, okay, it will push the receptor to the surface. And what will this receptor do? It will quickly suck in all the extra glucose, and so what will happen is this will lead to decrease in the blood glucose. What happens in insulin resistance? What happens in insulin resistance? In insulin resistance, what happens is, in insulin resistance, the insulin is not able to bring the receptor onto the surface, and that is why in type 2 diabetes it is insulin resistance, and it is the muscle cell which is not able to draw the glucose. Is it clear? So muscle has an effect on blood glucose, but not through glycogen, but through sucking of heat. So insulin resistance happens in your muscles, and that is why if you keep exercising, your your insulin resistance will be not there. Okay, your insulin sensitive will be there, and they can, you know, transport this receptors onto the surface, and whatever you eat will be transported into the muscle. So this is how, and this is a very important question. Okay, so let's look at, so is everybody clear about glycogen breakdown? So this is the major pathway of glycogen breakdown, which is in adults. There is another, yeah, one more important point which I missed is, a very favorite question is, which is the enzyme which is common to both glycogenesis, that is formation of glycogen, and glycogenolysis? And the correct answer is phosphoglucomutase. So this enzyme is common to both glucogen formation as well as glycogen breakdown. A very favorite question, and you have to remember it. Let's look at, what, there is a minor pathway. So as I told you, that is the major pathway. It is primarily in adult, but in children we have a minor pathway, and this minor pathway takes place in lysosome. Lysosome has a special enzyme called as acid maltase, and what it does, it will take the glycogen and convert it completely into glucose. Okay, and this is very important in your neonates, anyone less than 1 year or 1 month, especially there it is very very important. And deficiency of this enzyme deficiency of this enzyme leads to an important glycogen storage disorder called as Pompe's disease or type 2 glycogen storage disorder. I'm just giving a hint. Okay, and here the child generally dies within 6 months, because the glycogen will be stored in their cardiac tissue, in their all the other organs, and primarily due to the cardiac failure, the child will die. Okay, so Pompe's disease. Is everybody clear about the minor pathway? Is everybody clear about the minor pathway? Very good. Now let's look at regulation of glycogen. So how glycogen metabolism is regulated? Now there are two important regulation mechanism. One is basically your energies or substrate signal. Second is hormonal, which means if the you know uh, cell is in energy rich state, or there is high amount of glucose. Okay, or there are certain hormones which influence the glycogen level in our body. So these are the two regulations. Let's quickly look at each one of them. So let's say this is an energy regulation. So if ATP is more, glycogen phosphorylase will be inhibited. Okay, if glucose is more, glycogen phosphorylase will be inhibited. And if okay, so is it clear, everybody? And if glucose 6 phosphate is more, again glycogen phosphorylase will be inhibited. And the logic is very simple. If you have more glucose, you know, you don't want glycogen breakdown to happen. Glycogen phosphorylase we know is a breakdown mechanism. If you have more glucose 1 phosphate, you don't want glycogen to be broken down. If you have more ATP, you don't want glycogen to be broken down, right? On the other hand, glycogen synthase is just affected on glucose 6 phosphate level. Now, this has a very, very important implication. Imagine a situation. Imagine a situation when the glucose level is, low, but glucose 6 phosphate level is high. We will see this situation in various glycogen storage disorder. Okay, glucose 6 phosphate is high, but glucose is low, which means the blood glucose level is low, but glucose 6 phosphate inside the cell is, let's say, high. Ideally, the glycogen should be broken, but here what will happen? More glycogen will be formed, and this will lead to glycogen storage. Is everybody clear? Is everybody clear about this? So remember, and this is a very, very important point when I'm talking about glycogen storage disorder in some other lecture. Remember this, that glycogen phosphorylase is affected by ATP level, glucose level, and glucose 6 phosphate level, whereas glycogen synthase is only affected by your glucose 6 phosphate level in a positive way. Is this clear? Very good. Let's look at how hormones affect the glycogen metabolism. So there are two hormones which will lead to breakdown. One is glucagon and one is epinephrine. Now both glucagon and epinephrine has the exact same mechanism. So what is the difference? Look, glucagon, the main function is to increase the blood glucose, and we know that only liver can increase the blood glucose. Muscles cannot increase the blood glucose. So glucagon receptors are only in the liver. Whereas epinephrine, you have a fight or flight reaction. Okay, so in that both glucose should increase and your muscle should also be prepared for that fight or flight reaction. So epinephrine receptors are present both in muscle and liver. So this you have to remember. Otherwise, both have the same exact mechanism of action. Okay. So let's look at how they act. So basically, glucagon and epinephrine will act on G protein, and they will activate adenyl cyclase. What will this adenyl cyclase do? It will convert ATP into cyclic AMP, which act as a secondary messenger. Okay, and what it will do, the cyclic AMP will activate protein kinase A, and what will protein kinase do? It will phosphorylate enzymes, and the moment your glycogen phosphorylase is activated, it becomes phosphorylase is phosphorylated, it becomes active, and the reverse happens to glycogen synthase. The moment it is phosphorylated, it will become deactivated. So what is going to happen? So whenever glucagon and epinephrine is there, this is active and this is inactive, right? Let me again, right? Whenever glucagon and epinephrine is there, this is activated and this is deactivated. So glycogen breakdown will happen, but glycogen synthesis will not happen. And this is what we wanted when glucagon and epinephrine is in the system. Is it clear how glucagon and epinephrine acts? You should know the pathway. It will be helping you in your college examination also. Both glucagon and epinephrine will activate G protein, which will activate atenyl cyclase, which will convert ATP into cyclic AMP, which will activate protein kinase A, which will phosphorylate enzyme. The moment glycogen is phosphorylase is phosphorylated, it will become active. The moment glycogen synthase is phosphorylated, it will become inactive. Now, let me tell you a very simple concept, because this is like which enzyme is active in phosphorylated form and which is active in dephosphorylated form. You don't have to learn it. I'll tell you a simple concept that after this session you will always be able to answer it. Remember, all the catabolic enzymes, which means all the enzymes which will increase blood glucose, they will be activated in phosphorylated form. And all the anabolic enzymes or pathways, they will be active in dephosphorylated. Now let's see an example. Let's talk about glycogen phosphorylase. Okay, glycogen phosphorylase will break down glycogen. So catabolism is happening. It will activate in phosphorylated form. Let's talk about glycogen synthase. Glycogen synthase, it's an anabolic enzyme, which means it will be active when more glucose is there and anabolism is happening, glycogen synthesis. So it will be active in dephosphorylated form. Enzymes of cholesterol biosynthesis. Cholesterol biosynthesis is an anabolic process, so it will be active in your dephosphorylate. Enzymes of ketone body synthesis. Now, ketone body essentially synthesis, even though it is synthesis, but it happens when the body is in catabolism. So it will be active in phosphorylated form. Enzymes of glycolysis. Enzymes of glycolysis. What do you think? Now, glycolysis, remember again, it is a catabolic, glucose is being broken down. So it will be active in your phosphorylated form. So this is a very simple concept. If you remember it, you will be able to answer most of the question regarding which enzyme is active in phosphorylated and which enzyme is active in dephosphorylated. Similarly, fatty acid synthesis, anabolic process, all the enzymes will be activated in dephosphorylated state. Now let's look at how insulin actually influences glycogen metabolism. So in insulin, the first action is it activates phosphatase. What will phosphatase do? It will remove the phosphate, dephosphorylation of enzyme. As I told you, this is active in phosphorylated state. The moment phosphate is removed, it becomes inactive and this becomes active. So what will happen? More glycogen synthesis will happen. But insulin has another important mechanism which you should remember. TCA, and yes, they are independent. Very, very correct. Okay. TCA, TCA cycle and electron transport chain are not very, they lead to each other, but electron transport chain is different, TCA is different. Okay. Let's look at insulin, the second mechanism. So insulin, on one hand, it activates phosphatases, which will remove the phosphate and so inactivate this, activate this. It also does it will activate phosphodiesterase. So what does this phosphodiesterase do? It will convert cyclic AMP to 5-AMP, and remember this was the major secondary messenger for glucagon and epinephrine action. So there will be decreased in cyclic AMP and phosphorylation will decrease. Okay, so these are the two mechanisms by which insulin acts. There is a third mechanism. Now remember, all this insulin and glucagon is good for fed and fasting state. Muscle wants to break down, not dependent on fed and fasting state. So there is a cyclic AMP independent pathway which is present in the muscle, that is called as calcium calmodulin sensitive pathway. Here what happens, the calcium, whenever there is the muscle is increased calcium concentration, so there is a calmodulin sensitive phosphorylase, and what it will do, it will simply activate the muscle phosphorylases. Okay, so again it will basically lead to the breakdown of glycogen. Okay, so remember it is independent of cyclic AMP pathway. Again, let me explain you what happens in muscle. Okay, muscle does not want to regulate its glycogen level based on the fed and fasting. Basically, it wants to break down glycogen when the muscle is basically exercising. So what happens? So whenever the muscle is basically contracting, the level of calcium in the muscle will increase, and what it will do is it will activate calcium calmodulin phosphorylase. Okay, what will is it will do? Okay, it will basically phosphorylate your glycogen phosphorylase. So this is phosphorylated, it will become active, and it will also phosphorylase glycogen synthase. Now this is, once it is phosphorylated, it will become inactive. So what will happen? Glycogen breakdown will happen. So in, and this, remember, this pathway does not require cyclic AMP. So this is a cyclic AMP independent pathway, which is it clear? Right. It's a very good question. Now look, I'll answer you in terms of a, remember, let's say ketone bodies. Ketone bodies will also be forming acetyl CoA. Okay, and this acetyl CoA will go into TCA cycle. Okay, and glucose will also be forming acetyl CoA through pyruvate, and again it will be going into TCA cycle. So you see, in this it is a pure catabolic. The body is completely in a catabolic state. But in this, the body can be in an anabolic state also, which means it has excess of glucose. But TCA cycle runs in both. And electron transport chain, because TCA cycle will produce your hydrogen ions, so electron transport chain will also run. So it's not, TCA cycle has nothing to do with your anabolic state or catabolic state. Is this clear? Suppose even fatty acid breakdown is happening, it will form acetyl CoA, which will go to TCA cycle. So the relationship is not very linear in that case. Is it clear? Very good. So this brings us to the end. I have talked about, in the session today, I have discussed about some very important, things. So first I discussed about what is glycogen. Okay. Then I discussed about why is glycogen needed, because it gives us 16 to 18 hours backup of glucose. Like your power bank is there, na? Backup of glucose. And after that, glycogen is not existent. After that, gluconeogenesis will take. That we'll talk in fed and fasting state. Third, we talked about two major organs, liver and muscle, where glycogen is stored. Okay, and we talked about their differences. So there are three differences. I've talked in detail about that. Then I talked about glycogen structure, and that glycogen structure was basically 1,4 bond, which was a linear bond, and 1,6 bond, which was a branched bond. Okay, and I told you how to easily remember it. Then we looked at the steps of glycogen synthesis. Okay, all the steps remembered. Here I talked about activated glucose, a very important question, activated glucose. Then we looked at steps of glycogen breakdown. Okay, steps of glycogen breakdown. And then we talked about regulation. In regulation, we talked about energy signals or substrate signals like ATP, glucose, glucose 6 phosphate in detail. Then we talked about epinephrine, glucagon, and your insulin, how they influence. And the last thing we talked about is how in muscle we have a cyclic AMP independent pathway. So all these are very, very important questions. Thank you, Bio Ninja. Thank you so much. And again, my request is that I want to connect to all of you. I want to send daily updates to all of you, and if you like this session, if you like this, please connect with me on my WhatsApp group. There are only limited students. There's only high class updates on this group, so it's medicoapps.org/groups, and join. If you are a first year student, make sure that you join only first year group, because there only you will get anatomy, physiology, biochemistry update. If you are a second year student, join the second year update. If you are an intern, post intern, so join the correct group so that the update is specifically tailor made for you. Thank you so much, guys. Thank you so much. And as I will keep announcing my next class, live class, please come and do attend. Thank you so much. Take care. If you have any doubt, leave in the comment and I'll definitely answer.